Welcome everyone to Hyperacusis Research first webinar. We're excited to have everyone online today, and we are recording this session, so we will be able to provide this for future reference to watch again, and for those who are not able to participate live to be able to see it. So I'm excited today that for our first webinar, we have Professor Salvi, who's going to be speaking to us. Professor Salvi is a distinguished professor at SUNY Buffalo, head of the Department of Communicative Disorders and Sciences, where he heads up a lab that does a lot of very detailed animal model work for various types of auditory disorders, including to hyperacusis. And we're excited at Hyperacusis Research that this year Professor Salvi has volunteered to be one of our scientific advisors. You can see in the picture one of my visits to Professor Salvi. I'm Brian Pollard, the president of Hyperacusis Research, pictured on the right. And I've spent a few times at the lab with Professor Salvi, where he has generously been helping us, partnering both with some of their work as well as helping to direct the overall work for the types of research that's going to lead us to understand the sources and potential causes of hyperacusis, as well as ultimately finding a cure. One thing about today's session is that given the background of the type of research Professor Salvi and his team performs at SUNY Buffalo, it is more oriented on the science side of research, not very much on the clinical side. And while I understand a lot of questions will relate to the clinical side, please know ahead of time that he may not be able to answer some of those questions. Some of those will be probably more answerable in future webinars where we hope to have other types of researchers involved. I may offer some inputs from what I've learned from other researchers, while my background is not one of auditory research. I come from an engineering field, but I'm more than happy to share learnings, though that's not the equivalent for an expert like Professor Salvi. So for one detail on the webinar today, if you go to the next slide, we are attempting to be able to include questions and answers, and a lot of those have been sent ahead of time. And so the first portion of time will be an overall discussion of the work that is being done at SUNY Buffalo by Professor Salvi until about half the hour pass, and then we will go into a Q&A session with some of those questions ahead of time. I understand already that we have a learning that the chat feature may not be the best way to do this. It's actually the questions feature, so feel free to utilize either of those. And while we will, you can submit those questions throughout the seminar, we're actually going to hold off to the end to do the answering part of them. So as you have questions, feel free to go ahead and type them in. I'll keep the list, and we'll take as many as we can at the end of the session for today's webinar. And with that, I'll hand it over to Professor Salvi.
Thank you, Brian, for the nice introduction. What I'll be talking about today, to a large extent, is some of the work that we've been doing looking at the gain of the central auditory system. This is, when you talk about gain, it's kind of like the volume control on your radio. And one of the things that we've been working on for a number of years and have noticed over the years is that oftentimes when you have some sort of injury to the ear, the brain turns up its volume control. And we think this might be one of the factors that leads to hyperacusis. It's still a matter of debate, and there's much more work to be done. So what I'm going to do today is I'm going to give you a little bit of background information on hyperacusis. you'll see some of the questions that you have are also questions that scientists have. Another one is the relationship of hyperacusis to things like tinnitus and hearing loss. What's the relationship between these disorders? When we talk about hearing loss, I've often had patients call me up and they tell me they're 50 or 60 years old and they have perfectly normal hearing. And what I remark to them is you probably have some sort of hearing loss that's hidden because it's not measured properly, or you could have certain types of pathologies that might not show up on typical hearing tests. And I'll discuss some of the work that we've done on that. Another area I'd like to go into is our brain imaging studies. When you talk to your colleagues, your family members, and tell them you have hyperacusis or you have tinnitus, there's nothing that you can point to objectively. They simply have to rely on your verbal reports. Back in the 1990s, I was involved in some brain imaging studies, I think some of the very earliest, that showed evidence of where parts of the brain you could see tinnitus appearing, and you could also, there was evidence of hyperacusis-like behavior in the brain. And one of the things that I mentioned already is that hearing loss often causes the brain to become hyperactive. This is somewhat of a paradox because usually when you have a hearing loss, you have less information going into your brain. So one of the things that we think that leads to brain hyperactivity and might be central to developing tinnitus is inhibition. We think that many brains, as your ear gets damaged, one of the reasons the brain possibly becomes hyperactive is there's less inhibitory control. It'd be like driving a car without a braking system. You only could accelerate. Okay. Then I'm going to talk about we've just recently developed ways that we think are quite good for measuring loudness and hyperacusis in animal models. We'll discuss some of that work. And one of the ways that we can induce hyperactivity and we think hyperacusis in the brain is by, at least with animal models, is by giving a drug that you know as aspirin. But if you take this drug, aspirin, in really, really high doses, probably much higher than any of you take, you can induce hyperactivity in the brain, you can induce hyperacusis in animals, and you can upregulate stress hormones. So we think stress might be another contributing factor. So hyperacusis, in our minds, may arise from reduced inhibition in the brain. So let me talk a little bit about hyperacusis, what we know about it from the literature. If you look in the literature and look at the prevalence of tinnitus, or not tinnitus, hyperacusis, or loudness intolerance, about 9% of the population have these sorts of problems, including my wife. Patients with the primary complaint of tinnitus, if patients come in and they say they have tinnitus, and you ask these people if they have hyperacusis, about 40% will also show evidence of hyperacusis. But if you reverse this, if you ask people who claim they have hyperacusis, how many of them have tinnitus, almost 90% also have tinnitus. So people with hyperacusis typically have tinnitus. There was a really interesting study done by Gu and Melcher. Jennifer Melcher is in Boston. She's done some interesting work on imaging tinnitus and hyperacusis. What they did in one of their research studies I thought was quite revealing is they found some people that had so-called normal hearing, and these people claimed to have tinnitus. They brought the people in, and none of these people really complained about having hyperacusis, but when they carefully evaluated them about whether they had problems with tolerating loud sounds, a large number of the tinnitus patients who had no complaints of hyperacusis, when they were carefully evaluated, turned out that many of them also had difficulty tolerating loud sounds. So I think one of the problems when clinicians see patients, even people that come to see them, oftentimes patients don't have a way of verbalizing that they're different. Their loudness tolerance abilities are different from those of normal people. So my wife, for example, if you ask her if she had hyperacusis, she would say, no, I don't have that. But when I take the dishes out of the dishwasher, one of her common complaints is I'm smashing the dishes, I'm making a lot of noise, where in reality, I don't find the noises that I'm generating very loud at all. So she's very sensitive to loud sounds. And I think this is true of many patients. They know when they have tinnitus because when they're in a quiet room they can hear it, but they have no way to gauge whether they have these loudness tolerance problems unless they're really extremely hypersensitive to loud sounds. Okay. So when I was working on, I've done a lot of work on tinnitus over the years, and I've also done, in more recent years, focused on hyperacusis. About 4 or 5 years ago, I organized a conference on tinnitus, and I wanted to gauge how much work had been done on tinnitus. So I went on the National Library of Medicine. I looked up all the publications that had the term tinnitus in the title. And I made a graph that shows the number of publications with tinnitus in it versus the years that I looked. So you can see back in the 1950s or 60s, there were maybe 10 to 20 papers per year published on tinnitus. And then in the 1980s, you see an increase. And some of these were related to grant initiatives. Back in the 1980s, the American Tinnitus Association gave out their first grant. There seemed to be an upsurge in research on tinnitus. The Tinnitus Research Consortium gave out their first grant in 1998. There was a big upsurge in publications. And then in 2006, the Tinnitus Research Initiative gave out grants, and again, there was a big upsurge in publications. So more than 200 publications in about 2007, 2008. Now I want to draw your attention over here to the graph on the right side. This is a graph that shows the number of papers with hyperacusis in the title. And you'll see that back in the 1960s and 1980s, there were almost no publications. Nobody had done any work or very little work on hyperacusis. You get into the 1980s and 1990s, there were maybe 2 to 4 publications. And you get into the 2000 era, and the number of papers with hyperacusis in the title jumps up to 7 or 8. So there's a huge disparity in the amount of research on tinnitus. There's many more than there are on hyperacusis. The scales here go from 0 to 10 for hyperacusis. Over on the left, they go from 0 to 200. So patients often will contact me and they'll say, why don't we know more about hyperacusis? And my answer is, very few people are working on it. And usually the number of people that work on a problem are related to the financial benefits from that. So researchers work on research areas where they think they can get funding for their research. So one of the ways that you can get more research on hyperacusis is to promote funding in that. And you can do that in a number of different ways. You can do that through private foundations, or you can also do this by trying to get the National Institute of Health or the Department of Defense to promote research in these areas. Usually this requires going to your local congressman or senator and trying to get them to allocate research monies for these topics. Okay. So let me take you and start giving you a little bit of primer on the auditory system. The auditory system, where we think hyperacusis has its origins, is pretty complicated. It begins down here in the inner ear cochlea. Sounds go into your inner ear, and there are cells in the inner ear called sensory hair cells. They act like a microphone. They convert the sound coming into neural activity. That neural activity, in turn, is relayed out of the inner ear, the cochlea, into the brain, and it's relayed out by a group of neurons called the auditory nerve, the 8th cranial nerve. That nerve basically transmits that information into another processing center called the cochlear nucleus, located over here. It's in the brain stem. It is relayed up to the lateral lemniscus. Then you get up to the midbrain, another relay center, the inferior colliculus. Then it goes higher into another part of the thalamus called the medial geniculate body. And finally, you get up to the highest level of the auditory pathway, the auditory cortex. This is where we think high-level information like speech, listening to movies or music, where the information gets processed and interpreted over here. But you need all of these processing centers to make a full-formed auditory perception. Okay. If the slide I just showed you previously was only the auditory pathway, but the auditory pathway has connections to many other parts of the brain. When you hear a sound, for example, the sound comes from the right side of your body. You often turn your head in that direction. And the reason you're able to turn your head in the direction where the sound comes from is your auditory system is connected to other processing centers like your sense of balance, your sense of touch, your sense of feeling. And sounds not only evoke auditory perceptions, those auditory perceptions often are associated with emotions. So there's a part of the brain called the limbic system that's thought to assign emotional content to sounds that we hear. Sounds can be interpreted as being pleasant. They can be interpreted as being dangerous. They can be interpreted as being unpleasant. So your auditory pathway is connected to the things like the limbic system. The limbic system can evoke emotions or reactions to them. They can change your heart rate. If you hear a fire alarm going off, for example, or you hear an ambulance, a siren, that can activate your heart rate, get your autonomic nervous system going, and change your heart rate. So it would be a mistake to think of the hyperacusis as only involving the auditory system. The auditory system is connected to other parts of the brain that are really play an important role in how we interpret and respond to the sounds that we hear. Okay. So hyperacusis is often associated with damage to the inner ear. The inner ear for hearing is called the cochlea, and so we refer to hyperacusis often associated with damage to the cochlea. And over here on the left side of the picture, I have a scanning electron micrograph. It's a picture taking showing the apical surface of the sensory hair cells in your ear. These hair cells are characterized by a group of sterocilia on their apical surface. When these sterocilia are bent or deflected, they allow ions to come into the cells, and these cells then convert the sound activity into neural activity. So you need these cells to hear. The outer hair cells are thought to act like amplifiers. They actually enhance the incoming sound that reaches the inner ear. And these cells over here, the inner hair cells, are kind of hidden from view. They actually transmit the sound from the ear into the brain through the auditory nerve. They communicate directly with the auditory nerve. So if you're noise exposed, when you get old, if you're exposed to ototoxic drugs, one of the things that happens is these sensory hair cells are damaged. Over on the right side, is a scanning electron micrograph. You can see the stereocilia are missing on these cells over here. They're missing on these cells over here. So when a sound comes in, even though it's able to produce some vibrations in the inner ear, these sounds have these sounds that come in have no way of activating these cells. So even though the microphone is still present, the microphone is not working and can't transmit that information to your brain. So this is a very common cause of hearing loss, damage to the sensory hair cells. So I'm showing you a graph over here of a typical audiogram that probably many of you have gone to see an audiologist and an otolaryngologist. They test your hearing. The way they do this, they present sounds of different frequencies. This is a low frequency sound, 0.25 kilohertz. This is a high frequency sound, 8 kilohertz. So you can think of this as a piano keyboard. The right side of the keyboard's over here, high frequencies. The left side of the keyboard is over here, low frequencies. If we hit the keyboard, if we have, in order to hear a sound, if we have to hit the keyboard very gently or very lightly, we'd have a good threshold. Our threshold would be very low. Here these individuals have low thresholds. The sounds of only about 5 or 10 dB can evoke a response. And if you go to the mid frequencies, likewise, sounds of only 5 to 10 decibels are enough to evoke a response. And these people look like they have normal hearing, and they probably should have normal hearing because these are young college students between the age of 20 and 27. And they look remarkably normal, both the females in red and the males in blue. So what happens with these patients, this is a typical way we get our hearing tested. Oops. But one of the things, if you test these hearings of these subjects completely by testing above 8 kilohertz, you can test, all of us can hear when we're born up to about 16 kilohertz, you'll notice something that happens. These male students all begin to develop hearing loss. Their thresholds begin to rise, whereas the females have normal hearing at the high frequencies. So if we were to get somebody that was 50 or 60 years old, they could have normal hearing at the low frequencies, but more often than not, their hearing thresholds would be elevated at frequencies between 8 and 16 kilohertz. These are frequencies that typically never get tested when you go in to get your hearing evaluated. So many people will tell me they have normal hearing, but I think if I were to test them in my clinic or my lab, that most of them would have hearing loss, just like these young male students here. So this would be an example of what I would call a hidden hearing loss. It's present, but it typically goes undiagnosed because the audiogram is never completely tested. Now I'm going to show you another type.
One quick input, Professor Salvi. Someone just asked that you could speak a little closer to the microphone.
Okay. Okay, I'll try to speak a little closer.
Thank you.
Okay. This is another form of hearing loss that would be hard to evaluate in a patient, but it was a type of hearing damage that's caused by a drug called carboplatin. This is a drug that patients would take when they would go for cancer therapy. It's an anti-cancer drug. There's certain animal models that if you give this drug to the animals, they develop a peculiar type of lesion, and this is a lesion that damages the inner hair cells. So these cells in this single row over here are inner hair cells. Normally this row should be completely filled up with black cells, but you can see that many of these cells are missing here. They're missing here. They're missing here. And if you go and look into the outer hair cell region, all the outer hair cells are present. So what's peculiar about this type of lesion is that the lesion occurs to the inner hair cells, and it's a lesion of the inner ear that kind of thins out and kills or damages every second or third or fourth cell. Now let's see what happens if we test the hearing of an animal with this type of lesion. So this animal was tested. We measured its hearing threshold down here in the bottom. We measured the animal's hearing threshold before we gave the drug. Those are shown by the open circles. So the thresholds are pretty low. They're around 0 to 10 dB. And then we give the animal, my screen is acting strange here. Okay, we give the animal the drug. We test its hearing 2 months later, and the animal's hearing looks completely normal. Then we take the animal's ear out and evaluate it, and we find that this animal is missing about 50 to 60 to 70% of its inner hair cells. So this audiogram, which is kind of one of the gold standards for a hearing test used by audiologists and otolaryngologists, is very sensitive to damage to the outer hair cells. But when the inner hair cells are damaged or the neurons are damaged, it's a test that doesn't detect that. So these animals actually, with so-called normal hearing, according to our audiogram, actually has a huge lesion of inner hair cells and auditory neurons. So this is a type of what we call hidden hearing loss. This type of hidden hearing loss can be detected in other ways. One of the ways it could be detected is by people with these types of lesions, we believe, would have trouble hearing in a noisy background. Their hearing thresholds might be normal, but when you put them in a noisy background, like at a restaurant, they would have trouble hearing. Okay, so this is an example of hidden hearing loss. Now I'm going to show you examples of how you can measure abnormal brain activity in patients. This is some work I did in the 1990s using a technique called positron emission tomography. And this technique uses a radioisotope to measure or assess brain activity in human subjects. So the patients are given a radioactive tracer. In our case, we would give them water that has a radioactive oxygen molecule in it. These oxygen molecules give off gamma rays, and you can detect the gamma rays by these gamma ray detectors that are located around this person's head. And you can use this to assess how much brain activity is going on at different locations. So we did this in some patients that had tinnitus, and we think they had hyperacusis. So the pictures that I'm showing you here are from normal hearing subjects. We took these normal hearing subjects and we played a 2,000 hertz tone to them. We presented the tone at a high intensity, 80 decibels, SPL. We presented to their right ear. And when we looked at the brains to see where the activity was occurring, we saw a lot of activity in the auditory cortex over here, these areas in bright orange, red colors. So this is a part of the auditory cortex where you see the activity. You see it in these areas in the auditory cortex have names, Brodmann areas 41, 22, and 41. So this is what happens when you present a sound to a normal hearing subject. What we did then is we looked to see whether the activity in the brains of the patients that we had who had tinnitus, whether their brains responded more robustly to sound. And what we found when we presented the 2 kilohertz sound to the patients and we compared the patients' brain activity to people that were normal, we contrasted or compared them, we found that the tinnitus patients who probably had hyperacusis had more brain activity than normal subjects. So this is the area of extra activity that we see in the brains of our tinnitus patients who we believe have hyperacusis. We didn't actually test these patients and ask them. But this is interesting that these people with tinnitus, and remember earlier I told you about 80 to 90% of tinnitus patients, or a large percentage of tinnitus patients, have hyperacusis. So we think this is probably an indicator that these brains are hyperactive. You present the sound to them and there's more activity. Okay, so I've just shown you data from normal subjects, human subjects. We can do something similar to this in animals, but in order to do this, to measure the brain activity in an animal model, one of the things we can do is we can put electrodes into different parts of the brain. And in this example, we put electrodes down on the auditory nerve. We put electrodes into the middle part of the auditory system, the inferior colliculus. And we also put electrodes in the auditory cortex. And we presented sounds of different intensity. So here's some sounds to the auditory nerve. You can see you get a response pretty early after about 2 milliseconds. This is further up in the brain. You get a response a little bit later, around 4 to 6 milliseconds. And this response, which is coming from the auditory cortex, you see a response to around 10 milliseconds. So the information is being relayed from the auditory nerve to the inferior colliculus all the way up to the auditory cortex. We want to see what happens in these subjects over here. So this is a cartoon of the brain, shows you the auditory nerve down here. We can measure the electrical activity coming out of the auditory nerve. It's hidden from view on my screen, but normally when you increase the sound level, the response from the auditory nerve would go up. That's shown in the black line. But after we give these animals a drug, this drug carboplatin, it damages the inner ear. It damages the output, reduces the output of the cochlea. You can see the reduced output here in this blue line. So damaged ear has less activity coming out of the cochlea going into the brain. If you look in this part of the brain stem, the cochlear nucleus, you get the same sort of picture. Less activity is going into the brain of this ear that's damaged, from a damaged ear. But when you get up here to the inferior colliculus, you see that the activity actually can be higher than normal. And when you get up to the cortex, you also see more activity. So the paradox here is we damage the ear, we get less neural activity going from the ear into the brain. You would expect that if there was less activity going into the brain, that when you got up to the cortex, there'd be less neural activity here. But we see just the opposite. We see that at high intensities, the brain activity is actually higher than normal. So we refer to this hyperactivity that you see at high sound levels as an increase in central gain. It's like the volume control of the brain has been turned up. So when a weak signal comes in, the brain jacks up or increases the signal level over here. So we think this may have something to do with hyperacusis, because hyperacusis patients often have hearing loss. We think there's less activity going from their ear to their brain, but when that neural activity reaches the cortex, it could be much greater than normal. We've seen evidence of this in our imaging studies, in Jennifer Melcher, who's done imaging studies with hyperacusis patients, has seen something similar. So this is a nice demonstration in an animal model. Okay. So how could this why would the brain produce more activity when it's getting a smaller signal? So we started investigating this probably about 10 or 15 years ago. The brain, the central parts of the brain normally have strong inhibitory circuits in them. And one of the important inhibitory neurotransmitters is called GABA. And you can block the activity of GABA by putting a drug on called bicuculin. This will block inhibition. So this is the response of the auditory cortex of an animal model. We measure this, and then we apply this drug bicuculin to it. And one of the things you can see is the auditory cortex response gets about 2 or 3 times bigger. So that's telling us that the brain normally has a lot of inhibition controlling how much it responds. So what we did then is we did some quantitative measures of this. We measured the amount of the strength of the responses in animals that had normal hearing. We applied this drug bicuculin, shown in green over here. After we applied the drug, the negative part of the response, shown over here, gets much bigger, and the positive part of the response, shown over here in red, also gets bigger. So when we put the drug on, this response gets much bigger. If we do exactly the same experiment that we just did above over here, but we do it with animals that have been treated with carboplatin that are missing a lot of their inner hair cells, they have a very weak input to the brain, we apply this drug, and it's as if the drug has no effect. The inhibition's already removed. So we think what's happening in these ears that are damaged, the inhibition, the brain takes it away. It doesn't need it anymore because the signals coming in from the periphery are very weak. So applying the drug to a damaged ear has basically no effect because the inhibition probably has already been removed by our nervous system. So we think lack of inhibition is one of the things that may lead to brain hyperactivity. Now, one of the big obstacles to doing physiological or anatomical work and correlating with hyperacusis is you've somehow got to get an animal to tell you when things are too loud. So you can ask a human being when a sound is too loud by just telling you, when is the sound too loud? Put your hand up. And you can measure the sound intensities that are too loud for a person. But an animal is much more difficult. They can't respond to you verbally. But what you can do is you could retrain them to respond to a sound to get a food pellet. So we have animals that are they're food restricted. We feed them at a certain time of day, and they go in and they start a trial, and we measure how quickly they respond to a sound of various intensities. So here we have sounds that go from low intensities to high intensities. And what we do is we measure their reaction time. So if the guy's hungry and he hears a really loud sound, he responds very quickly. He runs to the table to get his food. But if the sound is very weak, it's near the animal's threshold, the reaction time is much longer. So you'll notice over here in these animals that are food restricted, as the sound intensity goes up, they respond faster and faster. So now we wanted to do is see if we could find any evidence of hyperacusis. So one of the ways we can induce tinnitus is by giving a drug called aspirin. Aspirin, the active ingredient in aspirin is sodium salicylate. So in our animal models, we give them a big dose of sodium salicylate that normally would induce hyperacusis, and we thought it would induce hyperacusis also. So we gave the animals this drug. Same animals, we measured the reaction time. Down here at low intensities, the reaction times are a little bit longer than normal, but as we go up in intensity, you can see the reaction times catch up to what they were before, and they actually become shorter than normal. So we think in this region over here, at higher intensities from about 60 dB up to about 90 dB, the animals are experiencing sounds that they perceive as much louder than normal. And we believe they're much louder than normal because the animals respond much more rapidly. So let me give you some perspective on these sound pressure levels. Normal speech is around 60 to 70 dB. The loudness discomfort levels for most normal hearing people is about 90. And people that have hyperacusis oftentimes have loudness discomfort levels that are down around 70, 75 dB. My wife's loudness discomfort level is around 75 dB if I play a sound around 8 kilohertz. So now we think we have a nice behavioral readout. We can tell when an animal has hyperacusis using this simple reaction time measure. This reaction time measure is also shown to work very well in human subjects. So it provides a nice objective readout. Let's see what salicylate does to the brains and ears of animals. So one of the things we can do is we can give animals a big dose of sodium salicylate. We can measure the response that comes out of the inner ear. This is called the compound action potential. This shows you how big the compound action potential is. This shows you the sound pressure levels. So before we give the drug, the inner ear gives us a nice big response, and the response goes up as we increase the sound pressure level. But after I give the animals this big dose of sodium salicylate, they get a hearing loss. So these functions are shifted to the right about 20 to 30 dB. They get a temporary hearing loss of about 20 to 30 dB. But more importantly, the ear produces less activity. It's sending less information into the brain. So this is very similar to carboplatin, very similar to what you would get with a noise-induced hearing loss, very similar to what you would get with an age-related hearing loss. So we're reducing the output of the cochlea or the inner ear. What happens when we get to the brain? So in this example, we put recording electrodes into the brain. We can measure the response from the brain to a sound, an 8 kilohertz sound. You can see the response is shown over here before we give the drug. Then we give the animal 300 milligrams per kilogram of salicylate. And it's very clear when you look at the response over here to the right, about an hour or 2 hours later, the response is almost doubled or tripled in size. So even though this drug reduces the output of the ear, the brain responds more robustly at high sound levels. Okay, I've shown you an area of the brain that's known to be involved in hearing. I told you earlier that the auditory brain communicates with other parts of the nervous system. One of the parts of the brain, called the amygdala, is an area of the brain that assigns emotions to things that we see, hear, or smell. It turns out the amygdala also responds to sound. This black line over here, dashed line, shows you the response of the amygdala to a sound stimulus, and it has a nice, robust response. What happens to the amygdala if we give an animal a high dose of salicylate? It induces hyperacusis and induces tinnitus. Well, that's shown over here in the red line. You can see the response has gotten much larger after we gave the drug that induces hyperacusis and tinnitus. And you can see quantitatively the effects on the right, where we have a graph of the size of this response before we give the drug, which is in black, and 2 to 3 hours after we give the drug. There's a hearing loss, so the function is shifted to the right. But after we give the drug, the amygdala is responding more robustly. So again, we see this hyperactivity arising in the central nervous system, even though the brain is getting less input from the ear. So one of the things we were wondering is, what is it that's driving up the activity in the brain? Is it correlated with things like stress? Well, it's hard to ask animals if they have stress or anxiety, but what you can do is you can measure things that are stressed. They're called stress hormones. So one of the stress hormones that the body releases is called corticosterol. You can measure this in a blood sample. So we treated animals with a high dose of sodium salicylate, and we wanted to see if it induced a stress response. And the way we did this is we took blood samples and looked to see if we could measure this stress hormone. So here on the bottom part of this graph is the amount of sodium salicylate we're giving. 0 means that's the control. 50 milligrams per kilogram, that's a very low dose. It would not induce hyperacusis. It would not induce tinnitus. But these doses over here, we know 150 and 250 milligrams, we know from our behavioral experiments that these doses would induce hyperacusis in animals, and they would also induce tinnitus. And you'll notice over here is that as we increase the dose of salicylate, the doses that induce hyperacusis and tinnitus, they also cause a huge upsurge in the corticosterol stress hormone. So one of the things we think that might be going on with sodium salicylate, why it's such a good inducer of tinnitus and why it's such a good inducer of hyperacusis, is it not only induces a hearing loss, but it also drives up the stress hormones. So this could be one of the cofactors that makes the central nervous system more responsive to hearing loss, makes the central nervous system more reactive. There are these stress hormones. So let me summarize. Many patients that have hyperacusis, many of them have tinnitus. If we look at their brains, or you look at their inner ears, many of them have hearing loss. Sometimes the hearing loss is hidden from view because we don't measure the full range of frequencies, or we don't use more sophisticated tests. But we know when we have some damage to the inner ear that one of the things that happens is the brain tries to compensate for the lack of input by turning up its volume control. One of the ways I think of this is if I have a radio station that I'm listening to and I travel out of town, my radio station signal gets weaker, and as the signal weakens, I reach over and turn up the volume control on my radio, and now I begin to hear the static, and sometimes the volume gets too loud for me to tolerate. We think the volume control in the brain gets increased because there's reduced inhibition, and the reduced inhibition makes very weak signals able to drive or activate the brain. So less excitation is needed if there's less inhibition. So we think this might be one of the mechanisms for hyperacusis. But really a lot more work needs to be done. I told you about the number of papers that were published on hyperacusis, only about 8 to 10 papers per year back in the early 2000s. If you look at cancer research or epilepsy research, there's thousands of papers published each year, maybe each week, on these topics. So one of the reasons we don't know very much is we just have had not much research done on hyperacusis. We think that developing animal models is one of the ways to promote research and get a better understanding of the biological basis of hyperacusis. So I'm going to stop here and try to respond to a number of questions that came in. I'll take them in on order. So Brian, are you okay with?
Yeah, one quick input, given that we've got about 10 minutes, is some of these may be similar to questions that folks have put in on the question feature. But because we have so many questions on the question feature, and I know we won't have time to get to all of these, I thought maybe instead of directly answering these first, we could, because these represent questions by and large by people who weren't able to attend today, and we could answer them separately, that maybe we can answer some of the ones that are online, and if we have time, we'll come back to these. Does that sound like that will work?
Okay, can I see the questions that have just come in?
Unfortunately, I don't know if you click on, did you ever find the question icon on the right? Well, I can just read them then.
Okay.
And we'll go by that plan. So one quick summary statement just that relates to a couple of questions, and I wanted to clarify. Several folks have asked about, is there a cure? Are we going to be talking about treatments today? And I think Professor Salvi may add a little bit to this, but one key point that I wanted to highlight is what you're seeing in today's discussion is what we at Hyperacusis Research believe needs to be the depth of types of understandings that we are needed for the auditory processes to ultimately reach a cure. And part of the challenge historically is there's been a lot of clinically focused work on treatments, which sometimes those treatments do make a big difference for a lot of patients, such as sound therapy. However, patients for whom those plans don't work are left hanging. And so it's a big emphasis of ours to go help drive this core research so that ultimately there can be a cure and a treatment that will work for everybody. And this additional depth takes an enormous amount of work, as you can see, a lot more funding, a lot more papers needed to be published. And so that's a key emphasis we have, is helping folks like Professor Salvi and teams like his to go to that next level. So
how about if I try
to go ahead.
How about if I respond to the, is there a cure for hyperacusis?
Sure, go ahead.
So many individuals call me up and ask about cures for tinnitus and hyperacusis, and we have a lot of patients that come into our clinic to get treated for tinnitus. Most of the patients that come in, actually, another complaint that they usually don't recognize is they have suffered from hyperacusis, and the conventional treatment in our clinic right now is kind of combination of sound therapy and counseling. And our clinicians tell me that the first thing that responds to of their problems, tinnitus or hyperacusis is the one that usually gets better the soonest. And a simple way to start is really getting a hearing aid. If you're 50 or 60 years old, you most likely have some sort of hearing loss. If it's not in the normal audiometric range, it could be above 10 kilohertz. So I usually tell patients, start off with a hearing aid. A friend of mine who has quite a severe hearing loss has a hearing aid. His hearing aid stopped working all of a sudden. He was kind of unaware of it. Went around with a non-functional hearing aid for about 2 months, and then got his hearing aid repaired. He put the hearing aid on. He said everything sounded too loud for him because he had been deprived of sound for about 2 months. And his audiologist said, just be patient, leave the sound where it is. In a month or two, or a week or a few weeks from now, everything will sound normal. I just had coffee with him yesterday morning. He was telling me about this, and I was thinking, this is a perfect anecdote to bring into the conversation today. I think sound therapy for many individuals works, but I think the way to get into it is you have to go into it gradually. If you get a hearing aid or a sound generator, you want the volume to be low to begin with and then gradually increase it. Brian, I think, raised a really important question. What do we do for the people where sound therapy doesn't work? And I don't have the answer for this. Part of it, I think, might be the counseling component of that, but I would say that there are a group of patients that, for both tinnitus and hyperacusis, that we really don't have very good treatments for right at the moment.
Right.
I think that's an area that really needs a lot more clinical work as well as basic science work.
Okay, so I'll just try to rattle off a few quick questions, and we'll go through, try to get in at least 5 or 6 of these. We have about 20 questions online, so I'll just pick some that are representative. So given that you talked about all of what was done for the animal model studies to give them salicylate, given that that can at certain dosage levels create ear damage, should people with hyperacusis avoid aspirin?
Unless you really overdose on aspirin, aspirin will not have much of an effect if you take just a few tablets a day. The doses that we're giving them are extremely high doses, and the reason people know that high doses of aspirin cause tinnitus really dates back to the era of rheumatoid arthritis, where the way patients would titrate their dose of aspirin to treat their arthritis is the doctor would say, keep increasing your dose of aspirin till your ears start ringing, and then when you reach that point, lower the dose, and that's your maximum dose to take. So we sort of discovered this almost by accident by treating rheumatoid arthritis patients. You would never take such high doses normally. So I don't think it's really a risk factor for most individuals. Having said that, there are a few individuals that are very sensitive to aspirin, but they're pretty rare. I know one research scientist in Rochester that's sensitive to salicylate. But you can do the experiment on yourself. Take a normal dose of aspirin. If it doesn't exacerbate your hyperacusis, you're probably okay.
Okay. Yeah, I understand we might have lost sound for a little bit, but I believe it's back now. So a lot of questions are relating to different types. I'll ask a representative question than a specific one. A lot of patients wonder about the nature of the risk to noise and whether or not they can incur additional damage from what is normally considered everyday noises. What are the recommendations related to, say 90 dB and above sounds? Should hyperacusis patients, for example, wear earplugs in places where there's concern about loud noise?
I think this is a bit of a dilemma for people that have hyperacusis. In most cases, I would say the hyperacusis patients should have some sound in their environment most of the time. That keeps the auditory nervous system busy, and it basically starts turning down its gain. But when the sounds get too loud, like you're saying 90, 95 dB, then you also get into the risk range for making the hyperacusis worse or even causing some hearing loss. So I think you want to avoid really very loud sounds, but you want to have kind of normal, you want to try to have some normal everyday sounds in the environment. You don't want to deprive yourself of sounds. So I'll mention an experiment that was done by Craig Formby maybe 15 years ago. He took some individuals, he put earplugs on them, made them wear them 23 hours a day. Before they wore the earplugs, they had to tell the investigator what sounds were too loud. The patients would say the sounds get too loud at 100 decibels. After they wore the earplugs for a month, sounds around 92 dB were too loud. So their sensitivity, their loudness sensitivity had actually sort of decreased, meaning they became more hypersensitive to sound. If these patients or individuals wore sound generators for 23 hours a day for a month, what happened is just the opposite. They could tolerate much louder sounds before they became uncomfortable. So that's really a good example, scientific example of how your sound environment can change what you perceive as being too loud or too quiet.
Right. Okay. So I think we're going to have to stop now. We have a lot more questions, so I will copy these down and send them over to Professor Salvi. We'll attempt to answer as many as we can with our Facebook posting, eventually get it onto our main site. I really appreciate Professor Salvi's time today and all of the participants. A lot of great questions. Again, we hope to be able to build on this webinar to do additional webinars in the future. Perhaps we will have one more dedicated to question and answer since we got so many. And I just want to, again, thank everyone for participating. One final comment is, was asked to, how can we get more research? The number one way is to get initial funding. Supporting funding for our organization is one way to do that. We are a fully volunteer organization and try to turn as much of the funding as we can right directly into grants to people like Professor Salvi's team in partnership with the Hearing Health Foundation, who's really been a great partner in helping us to get grants administered. And we have one upcoming this next year where we're going to fund a student to do some work dedicated to hyperacusis. So that all helps the NIH see the level of interest that the community has. And as that level of interest grows and more people are getting at least small grants, that enables larger grants to be created by the NIH to help the programs grow overall. So thanks again, everyone, and we will hopefully have this recording uploaded and provide a link for that, and we'll get as many other questions answered as possible. Thank you again.